Okay, so I think we may have a couple of people joining as we go here, but I think we're ready to get started. Sorry again, everybody, about the confusion with the Zoom. We had created the breakout links under the same Zoom account, and Zoom is constantly changing their policies about what's allowed and what's not allowed there. So it turns out we were only allowed to have two rooms, but it looks like everybody who was interested from our core group has found us successfully. Um, so thank you again for your patience, and we'll go ahead and get started here. So I will give Megan a very quick introduction. So thank you, Megan, for agreeing to come talk about your CompuCell work with us today. I know that our participants will appreciate it. Um, so Megan earned her BS in Biomedical and Health Sciences Engineering from North Carolina State University um, and the University of North Carolina at Chapel Hill in May 2020. Sounds like it was a joint department. She's currently a fourth-year PhD candidate in the multi-scale muscle mechanophysiology lab at the University of Virginia. Her research focuses on using experimental and computational approaches to understand how changes in the skeletal muscle tissue microenvironment impact muscle regeneration. I believe that's what she'll be talking to us about today. So just as a quick reminder, before we get started, this session is being recorded. We are not currently live streaming, but this will be posted to YouTube for viewing in the future. So just so everybody is aware, the session is being recorded. And I think with that, I can turn it over to Megan to get started.
Cool. Thank you so much for the introduction. I'm really excited to get to talk a little bit about some of my PhD work. It's also really cool because back in like 2021, I was in the CompuCell3D Workshop, kind of where everyone is now, just learning all of the basics. And a lot of the work that I'm going to be talking about, actually, like the framework of it was built during the hackathon. So it's cool to kind of come back and share where everything is at now. So with that, I'm going to be talking about using CompuCell3D to predict how microenvironmental changes impact muscle regeneration outcomes. So I'm sure, as many of you likely already know, muscle regeneration is really essential for everyday life. So if you think about breathing, you're using diaphragm muscles, obviously walking, running, any sort of physical activities using muscles. And so if we kind of like look at the breakdown of this, so we have our muscles that kind of within this muscle belly, we have these individual fascicles that hold these muscle fiber cells, and within those we have these muscle myofibrils. And so if we kind of take that muscle fascicle and cut it and look at the cross section, we can see these individual muscle fibers that sit within this extracellular matrix. There are also capillaries that are bringing nutrients, blood into that environment, as well as these satellite stem cells, which are essentially precursor muscle cells that can help them grow and also repair. So we also know that throughout daily living, as well as in many other ways we can injure muscle, and it has to have these regenerative abilities, which it's remarkably good at. And so to dive a little bit into that and kind of the whole regeneration cascade, so if we are to look at that cross-section of muscle, we have these healthy muscle fibers, and then upon some sort of muscle injury, like a muscle tear, a muscle strain, we get some acute damage, which is called necrosis, so that's these damaged fibers. And we have this inflammatory phase where the neutrophils first flood in to essentially eat away at some of this necrosis to help clear that damage. And then following that, in this kind of second stage of inflammation, we have pro-inflammatory macrophages. They kind of come in and clean up whatever is left over from these neutrophils and kind of assist them to clear up necrosis. And then also during this phase, we have fibroblasts that move in, and their role is really to rebuild up the extracellular matrix that is damaged during injury. And so they will secrete collagen that helps rebuild up this extracellular matrix so that the fibers can then be placed down to repair. And then we also have these satellite stem cells, and initially they are quiescent when it is a non-injured muscle, but following injury, we have a variety of different signaling factors that cause their activation. Following this, we have the remodeling phase. So during this time, we see a shift from the pro to anti-inflammatory macrophages. And so these macrophages are really going to start secreting factors that transition the microenvironment to help the satellite stem cells differentiate so that they can begin to fuse to repair fibers. And then following this, we have the regenerative and muscle growth phase. So these differentiated satellite stem cells will fuse to damaged fibers to help them regrow, as well as they can fuse to each other to form new fibers. So it's a fairly complicated cellular process. There's a lot of moving parts, and it gets a little bit more complicated because there's more going on within this extracellular matrix that has to be accounted for. So the ECM is made up of these collagen fibers, and so it's essentially this scaffold that kind of can help hold a lot of these fibers in place. And it's also where all the cells are moving around, as well as where the signaling factors are going to diffuse and decay. And so these collagen fibers make up these collagen bundles, which are essential for that structure. And as I mentioned, we have signaling factors, so growth factors and cytokines that are diffusing and decaying throughout this environment that are essentially dictating how all of these different cells are behaving. So our goal was to build an agent-based model to predict regeneration outcomes with altered cytokine dynamics. And so this kind of gets broken down into a couple of different parts. So the first of which, we wanted to develop a computational model of muscle regeneration that includes cellular and cytokine spatial dynamics, as well as a microvascular environment. We wanted to calibrate the model to capture cell behaviors from published experimental studies, as well as validate additional model outcomes by comparison with other experimental studies. And then lastly, we wanted to conduct in silico experiments to predict how altering different cytokine dynamics could impact muscle regeneration. So with that, I'm going to jump into a couple of example agents just to kind of show some of the dynamics that are occurring within the model. So for our satellite stem cells, kind of as we mentioned, upon initial injury, there is an influx of HGF, which is this damaged cytokine that causes the satellite stem cells within the model to activate. A different subset of cytokines at correct thresholds will then cause those cells to undergo cell division. Following that, similarly, we have a different subset of cytokines that can influence those cells to undergo differentiation. And then once those differentiated cells are nearby a damaged fiber or another differentiated cell, they can fuse to help form a fiber. And then we also have our inflammatory cells, such as our neutrophils. They migrate towards areas of necrosis to phagocytose it, and then as they're phagocytosing, they secrete MMP, which is helping them break down that damage. And then following this, they can undergo cell death, where they essentially leave the simulation. And then we have our macrophages. They chemotax or migrate along an MCP gradient, and that allows them to phagocytose damage, as well as they can remove apoptotic neutrophils. So this is kind of like a helpful visualization to see some of the rules that are occurring within the model. But it gets a lot deeper than this. So if we kind of break down and look at all of the individual rules that are tied back to literature sources for each of these dynamics, there's a lot of stuff that kind of goes in on the back end to support all of these different dynamics and start to define different parameters for those rules within CompuCell. So this is not the first model of muscle regeneration. There have been many before that have kind of helped set up some of these rules. So the first of which, it was built in NetLogo, and it was really focused on kind of how muscle is adapting. But it had really limited diffusion dynamics as well as simplified cell dynamics. We also, from our lab, have a muscle regeneration model that was built in Repast, and it focused on a muscle disease state called Duchenne muscular dystrophy. And then we've also had kind of an extension of that one that focuses on volumetric muscle loss, also built in Repast. But neither of these included spatial diffusion of cytokines or spatial dynamics of inflammatory cells. And kind of as we talked about earlier, so many of those spatial dynamics within the microenvironment are really crucial for understanding regeneration, that it was something that we wanted to study further. And so because of that, we kind of turned to CompuCell3D because it allowed a lot of these intricate spatial cell dynamics as well as cytokine diffusion in an easier manner than a lot of other agent-based modeling platforms. So for our model, we had a variety of different complex cell behaviors that were modeled spatially. So all of our inflammatory cells that we've kind of walked through before. So we have our neutrophils as well as monocytes, which are able to transition to M1, so pro or anti-inflammatory macrophages. We also have the full satellite stem cell lineage. So the satellite stem cells are one cell type within CompuCell3D, but they each have a different dictionary property that can dictate what state or what portion of that lineage they are. Similarly, we can have different types of fibroblasts, and then we also have our microstructure elements, so ECM, fiber, lymphatic vessel, and capillaries. And then we have a variety of cytokines that are constantly diffusing and decaying throughout the simulation. And so example behaviors for these cells, a lot of them can proliferate, differentiate, undergo quiescence or apoptosis. The cells are also secreting a variety of these cytokines, and then with each behavior that occurs, they are removing the cytokine that initiated that behavior, as well as migration and activation. So now to kind of go into initialization. So all of the spatial characteristics within the model are defined from histology. So we started and we took a cross-section of muscle tissue and we stained it with H&E so that we could see the fiber and the ECM borders. And so then we took a subset of this immunohistochemistry image and then we read it into MATLAB so that it could define what elements were fiber and what elements were ECM. That then got read into MATLAB and generated a CompuCell3D file, and we would assign the microstructure elements, so our ECM, our lymphatic vessel, capillary, and muscle fibers. This then got saved as a PIF, which is like kind of a CompuCell unique initialization. So then each time when the model initializes, you don't have to redo the whole reading it through MATLAB and running the initialization file. You can just load in the PIF, which was super helpful. And then the user is able to define a preset amount of damage, so some percentage of necrosis that occurs. And then we also have a randomized placement of baseline cells. So these are essentially like the resident cells that are there pre-injury. And then the kind of unique thing with our model setup is it's essentially, when we started, it was like a 2.5D model where we had our fiber layer on one, and then we had a wall separating the other like inflammatory cells so that they wouldn't like move back and forth. It's since been kind of updated and we've refined a lot of the like CompuCell contact energies so that we could have it without like a wall plane and have more interactions between the two. And then from there, so the damaged fibers secrete HGF and TGF at model initialization. And then that kind of gears up the simulation to go through all of the step-uples. So another aspect of the model that was fairly new is this incorporation of microvascular growth and remodeling. So we know that we have these fibers, and then we have in orange these capillaries that are kind of running along the fibers. And so that was incorporated into the model, essentially using literature-defined capillary to myofiber ratios in terms of how many capillaries would be placed within the model. And then they're able to bring in a variety of cell types as well as cytokines. And then following damage, capillaries that are next to areas of damage are also damaged and have to undergo angiogenesis to get repaired. And then we can also get new capillary formation within the model. And then they're able to bring in additional nutrients to that tissue microenvironment. Last piece kind of here, we also incorporated some dynamic ECM properties. So if we kind of remember back to earlier in the talk, we know that ECM is really crucial. It's essentially the scaffolding that's holding everything in place. And so the fibroblasts throughout the simulation are secreting collagen. In CompuCell, this is essentially done by having like an ECM dictionary property for the collagen density. And so throughout the simulation, the collagen is changing. And we also wanted to account for this for our cytokine diffusivity. So for each time step within CompuCell, so each MCS, we calculate what the collagen density is for that tissue microenvironment. We then account for that and use this formula to recalculate the diffusivity of our cytokines, and then that gets updated so that we're actually accounting for changes within the ECM as these cytokines diffuse. So with that, we've kind of covered all of the initialization steps. So the next thing that occurs is we get recruitment. So our neutrophils are recruited to the simulation proportional to the number of damaged fibers. Monocytes are recruited proportional to the cytokine MCP, and then satellite stem cells are recruited proportional to HGF and MMP while being deterred by TGF. And so following that recruitment, so each of these are essentially kind of representing a steppable within CompuCell. So within our neutrophil steppable, they check if they are able to undergo apoptosis based on a certain time that they've been in the simulation or how many things they've phagocytosed. If so, they leave the simulation. Otherwise, if they are near necrosis, they phagocytose. If not, they migrate along their gradient and uptake it as they do so. Macrophages get a little more involved. So, like I said, there's a variety of different phenotypes that they can be. So, starting as a monocyte, they can check if there's enough cytokine within that location of the monocyte to transition. If so, they can transition into an M1. If both M1s and monocytes can phagocytose damage, whereas M2s can proliferate as well as secrete anti-inflammatory signals. And then satellite stem cells are another one that are pretty involved, but we've kind of already talked through a lot of these behaviors. So they can differentiate both symmetrically and asymmetrically. So essentially meaning if it's going to proliferate and add another satellite stem cell, or if it's going to proliferate and add a differentiated satellite stem cell. And then we also can have apoptosis as well as fusion to another differentiated satellite stem cell or a damaged fiber, all of which is kind of tying back to if that collagen is able to support that fusion behavior. Then for our fibroblasts, they can be activated, they can differentiate, as well as divide, undergo apoptosis. And then following all of these different cell behaviors, we start to see microstructure changes. So, recalculating that diffusivity rate, as well as having the healthy fibers secrete their proper cytokines. And then if there is a proper angiogenesis signal, it can help damaged capillaries be able to reperfuse or can generate new capillary sprouts. And so then this starts again. Every MCS, we go through each of these steppables. And so kind of what this looks like in CompuCell. So we have our muscle cross-section, we see clearance of the damage, followed by fusion of new fibers to eventually kind of repair this cross-section of muscle tissue. And so we can also look at how the cells are migrating throughout this. So we saw inflammatory cells, and then division, differentiation of these satellite stem cells, and then how they're fusing to those fibers to help repair it. So you can see it again, all of the different migration, as well as we're kind of keeping those cells within the ECM, kind of how you would see within a biological tissue. Then we can also look at the cytokines and how they're changing throughout the simulation. So HGF is most prevalent at the beginning because it's this major damage signal, and then it diffuses and decays across the simulation, but it's also getting secreted and removed as cells are using it to initiate certain behaviors. And it gets a little bit more complicated as you throw in all of the other cytokines that are occurring throughout the simulation and all of their different, diffusion, decay rates, and usage by the different cell types. So yeah, the simulation represents like a 28-day period, and so we can kind of see both visually, but also get a variety of other quantitative metrics to explore with these simulations. So that kind of talks through the model development part. So a couple of takeaways that I learned from developing the model is that, from just a biology standpoint, there are things that seem to make sense. So for example, these satellite stem cells, we would think, oh, well, they're super beneficial for muscle regeneration. So obviously, if we just have a ton of satellite stem cells, that's going to give me the best fiber recovery. Well, we can see with the model that there's still too much of a good thing. And if you just spam the simulation with satellite stem cells, it doesn't really benefit recovery. There's a threshold where it's no longer beneficial. And so we kind of learned that through the development in terms of testing different parameters for that. Another takeaway was that forming biological rules is often not as simple as initially suspected. So there are certain things that seem obvious just from biology. So, for example, fibers don't merge, and that would make sense from biology. But from a computational standpoint, it's less obvious in terms of how to keep those dynamics across simulations as you incorporate more complexity. Another aspect of this is that translating cell behaviors into code takes a lot of trial and error. So there are a lot of things that aren't defined, either because they can't be measured or because they're an abstract computational parameter. And so there's a lot that goes into kind of getting the biology into a translatable code that can be used. And then the last piece of that is that there are a lot of unknown parameters that add another layer of complexity into model development. So there's always going to be something that you can't just look up the number to put in, and you'll have to kind of iterate on that, which can vary in complexity depending on how many unknown parameters there are. So then I'm going to jump a little bit into the calibration scheme that we used. So we used a tool called CaliPro, which is a recently published and available tool. And so essentially you start by taking your model, experimental data that you want to calibrate to, and all of your unknown parameters. And then you sample that unknown parameter space. So you start by kind of constraining what are the feasible bounds for those parameters. So you can determine that if you have a somewhat of a biological idea or using pilot testing with the model to see what would break the model. And so you can then do Latin hypercube sampling or other preferred sampling techniques to generate a variety of different parameter sets to test. Then you run a ton of simulations in CompuCell with all of these different parameter sets, and you set some sort of pass criteria for these different parameter sets. So for example, I calibrated to cross-sectional area. So that was one of the metrics that I started to use for my criteria. So I would only accept parameter sets that reached within a certain threshold for the experimental data for cross-sectional area. And then once you kind of narrow in on your criteria, you can then narrow in on your parameter space using either a highest density region or an alternative density subtraction. And so the highest density region is just taking the highest density of the runs that pass that criteria, whereas alternative density subtraction takes the ones that pass and the ones that failed and kind of subtract them to then narrow in that parameter space. And so from that, you get a new set of ranges and you kind of reiterate over that until you have reached some final termination criteria where you are happy with the state of your model's predictions and how it aligns with experimental data. So kind of thinking back to our flowchart of the satellite stem cell dynamics, you can imagine there are a variety of different roles where we have unknown parameters. So for example, how much signal does it take to activate a satellite stem cell, or how much collagen is needed for a satellite stem cell to be placed, etc. So like all of these were unknown parameters within the model. And so this is kind of showing a ton of simulations that we ran. So we did Latin hypercube sampling, we ran all of those parameter sets, and each line is a different run, and then the dots are representing the range of the experimental data as well as the average. And so then we use that criteria to constrain ones that were within that acceptable experimental range. Doing that, we could look at our alternative density subtraction to then narrow in some of the parameter space. So this is just showing a couple of example plots of that. And then we can also use partial rank correlation coefficient, which is essentially just a sensitivity method to look at how the different parameters are influencing outcomes across the span of the simulation. So this is showing across the different time points if that parameter is positively or negatively correlated with cross-sectional area recovery. And that's kind of just used to make sure that we understand that our parameters are doing what we expect them to do and not, non-sensitive, or if they're too overbearing on outcomes. And so with that, we had 52 unknown parameters. We did 4,800 parameter sets, so over 14,000 simulations were run because we did these in triplicate. And so this is just kind of showing the scheme of how this was done. So we took our parameter ranges, did Latin hypercube sampling, that went into CompuCell. Those outputs went into MATLAB to run it through CaliPro. And then we would test it with our calibration data and our set pass criteria, and then iterate until we were happy with where our outputs had landed. So we calibrated to muscle cross-sectional area, as well as satellite stem cell and fibroblast cell count. The reason that we chose these is because cross-sectional area is our major output. It's determining how quickly and how much of the muscle has recovered, so we knew we had to include that. And then the satellite stem cells and fibroblasts are also influential to the cross-sectional area outcome, and they're a very linked cell type, so we wanted to make sure that both of those were incorporated. And so you can see that for a majority of the experimental time points that the model was hitting within the standard deviation of the experimental data. So once we did that, we then took separate data and separate model outputs, and we compared them just to see would the models still align. And we found that it actually did. So the neutrophils and macrophage cell count were hitting the experimental ranges, as well as our capillaries per fiber area were also within experimental ranges. So this gave us confidence in the model's ability to kind of replicate a lot of the experimental data that had already been published. So then we wanted to kind of take this one step further. So we ran a variety of different model perturbations. So we were starting to play with different input conditions, simulating different experimental conditions that had already been tested. So one of which was depleting the microenvironment of neutrophils. So we did this by lowering the parameter for neutrophil recruitment proportion and found that it aligned with experimental data, decreasing cross-sectional area recovery. We also tested an anti-inflammatory nanoparticle, and while we didn't see the same significance that was seen experimentally, we did still see the decrease in M1 and increase in M2s. We tested IL-10 knockout by adjusting our diffusion and decay parameters and saw the decrease in cross-sectional area that was seen experimentally. We also tested hindered angiogenesis by upping the threshold required for those capillaries to undergo angiogenesis, and as seen experimentally, saw hindered cross-sectional area recovery. We also tested a variety of VEGF injections, which I'll touch on a little bit in later parts of the talk. We also did an MCP knockout, which resulted in a decrease in cross-sectional area recovery as seen experimentally. We also did a TNF knockout, and this one actually did not align with experimental data for cross-sectional area, but did align for the satellite stem cell differentiation in monocytes. And lastly, we also did a macrophage depletion, and we saw that that also decreased cross-sectional area data, aligning with experimental outcomes. So diving a little bit deeper into the VEGF perturbation, so with this, we looked at a variety of different levels of VEGF injection. And so VEGF is really influential for capillary recovery. And so we wanted to see if delivering different levels would influence cross-sectional area recovery, as well as if we were to prevent angiogenesis from occurring, how that would impact other aspects of regeneration. And so what we saw was that cross-sectional area was fairly similar across all of the injections, but that the extra high injection was not more beneficial than the high injection. And we also saw that for the hindered angiogenesis, that we saw upregulation of that HGF damage signal throughout the simulation. And we also saw that with the high injection of VEGF, we actually had an upregulation of fibroblasts, which could be contributing to the lack of increase in cross-sectional area recovery with that extra high condition. Another aspect for each of the different knockout conditions, we wanted to see how an individual knockout of a cytokine was influencing all of the other cytokines within the simulation. So for example, when we just removed MCP1, we saw that there were kind of this cascading temporal impact on all of the other cytokines. So the VEGF concentration within the simulation at day 28 was upregulated, so that dark red is indicating a great increase from baseline, whereas the purple is a decrease. And so we can see that it wasn't like a consistent trend of an increase or a decrease, but it was actually temporally dependent, even with one singular knockout. And we could see similar effects with TNF alpha knockout. So just knocking out that one cytokine caused great increases in TGF beta at the final time point. Um, and then for IL-10 knockout, we saw some interesting dynamics with TGF beta being decreased midway as well as at the end, whereas like MMP9, which we know is breaking down the ECM, was upregulated at day 7. And so this kind of got us thinking that if we could do like some other perturbation for these cytokines, how we could really leverage these cascading impacts to our advantage to get increases in cross-sectional area recovery. And so to start investigating this, we looked at partial rank correlation coefficient to kind of get a better idea of how altering the decay and diffusion properties within CompuCell would influence these different outputs. So we could see with HGF decay, increases in that parameter cause decreases in cross-sectional area pretty much across time. So the black dots are representing when it was a significant MCS or time point. We could also see which decay and diffusion changes were influential for satellite stem cells, as well as into macrophage count, and then start to kind of pick away the dynamics that were beneficial and leverage those to start making predictions on potential therapeutics. And so using Latin hypercube sampling to kind of sample the decay and diffusion parameters and then looking at those partial rank correlation coefficients, we put together this table, and it's showing kind of like highlighting the most influential time points and how those different decay and diffusion parameters were either positively or negatively correlated with certain model outputs. And so taking this, we then looked at some different correlations to see how different cytokine concentrations were correlated with different model outputs. And so we could see some trends were definitely stronger than others. So IL-10 concentration had a strong correlation with M2s, and VEGF had a strong correlation with the number of fragmented capillaries. And then we could also look at some interesting dynamics, like TNF had this bell-shaped curve with fibroblasts. And so now that we kind of had a good understanding of things that were correlated, as well as the different aspects that were beneficial for different metrics and how the decay and diffusion parameters were tied to that, we then wanted to test these individually as well as in combination. So we started running different simulations where we would just change one decay or one diffusion parameter that we knew had been beneficial when we did a full sweep of these parameters. And so we found that overall, all except for the MCP1 decay change resulted in enhancements in cross-sectional area recovery. But the really cool thing that we saw here was that when we did them in combination, that had a greater increase in cross-sectional area recovery than any of those individual parameter changes. And so we can also, now that we have all these other metrics from the model, we can gain new insight in terms of what other metrics could be contributing to the cross-sectional area increase. And so some of the interesting things that we saw was that the fibroblast count had a much higher peak, but it was a narrow peak, indicating that it could be helping the ECM repair more quickly, so then the satellite stem cells can place more rapidly, but not being a prolonged fibroblast state that would cause fibrosis. We also saw that the peak and the amount of satellite stem cells just generally over time was higher with this combined cytokine delivery, which is pretty cool to see that putting these all in combination kind of had this additive effect. So with that, kind of going back to our question in terms of how do different modifications to the tissue microenvironment impact regeneration outcomes, we found that varying VEGF injection doses showed increased cross-sectional area recovery up to a certain threshold, and then beyond that, further improvements stopped. We also found that with these cytokine knockout simulations, that removing one cytokine triggered this really complex cascading temporal impact on the system. And then we found that relationships between cytokines and cellular outputs had these nonlinear effects, and that combining the cytokine alterations have a stronger regenerative effect than any of the individual cytokines. And so this is published, and we also have our code publicly available, as well as instructions for kind of getting things initialized and what additions you have to add to CompuCell in terms of different packages and things. So happy to talk through any of that, as well as thank my lab and the CompuCell3D collaborators that I met back when I was in the workshop. We continue to work together. This is actually us at a conference that we went to like a year and a half after we met at the workshop. So it was really cool to kind of get to meet them in person, and like we still are working on this model in future publications that are working on different injury types and additional hormones in the microenvironment. So that, I'd like to thank you. I wanted to kind of show the glow up of the model. This version over here is kind of what we left. The left version is post hackathon, and then we've iterated on it for years now. So you can kind of see the more finalized, published, polished version. But yeah, so I'm happy to answer any questions about the process of model development, the perturbations, future work, as well as if anyone wants to walk through the steppables, I'm happy to pull up the code and just talk through like the structure or anything like that.
Okay, great. Thank you, Megan. So we've actually got a couple questions in the chat. I was going to ask, that conference was at SMB 2023.
Oh yeah.
Okay. Yeah, I was there too.
Oh yeah. Oh yeah. Fun campus.
But yeah, it was pretty
Yeah.
nice. All right, so yeah, we can look at the questions in the chat, and then if anybody has other questions, we can.
Yeah. So I'd see a question on, does the order of the steppables for each cell type matter? So yeah, that's a great question, and yes. So there are definitely some strategic like methods for how each steppable is organized. So within the like separate Python file that organizes the steppable, I have it pretty much set. So like, we obviously have initialization at the start, and then I go through each cell type. So like our inflammatory cells, then satellite stem cells, and fibroblasts. So it's a little bit linked. I don't know how much of an influence like changing one or two, like if I change like neutrophils and macrophages, I haven't tested to see how that would like alter. But definitely having some of the like initialization is required prior, just in order for that setup to occur for the next steppable. Um, and then, so for the CSA calibration, would that threshold be at a certain time point in the simulation or across the simulation? For example, were you looking for parameters that gave you 80 to 95 CSA recovery by 28 days? Also a very good question. So when we were doing CSA calibration, as I kind of said, it was like a pretty iterative process. So like the first criteria was very like basic. It was pretty much making sure that our like cell counts didn't explode, like making sure that things were like biological, just narrowing out parameters that were not feasible. And so from that, then I started to get a little bit more strict. So we had those different time points for CSA. So then I started using each of those time points and saying that the like cross-sectional area output from the model needed to be within like 2 times the standard deviation from the experiment at each time point. And then with each iteration would kind of like narrow that multiplier in more and more until it was just within the standard deviation. And that was used as our like termination criteria. So it's best to do that because when you're calibrating, you don't want to like narrow in your parameter space too quickly. Otherwise, you have to essentially backtrack, and that's never a fun time. Okay. And then does the lymphatic vessel get damaged when you initialize necrosis? If it does, how does that affect regeneration? That is also a super fun question. So our current model does not account for like lymphatic vessel damage. There is literature to support that like when muscle gets injured, that is also can be a consideration because the lymphatic vessel getting damaged would obviously influence like drainage of excess cells and cytokines. So it's something that definitely future work could incorporate, and that would be a cool addition to the model. The other aspect that would be really interesting is looking at like injury location, and like currently you wouldn't really be able to represent like the impact of an injury near versus away from the lymphatic vessel because it's not accounting for like an injury near the lymphatic vessel would probably it would damage the lymphatic vessel. But if it had that new incorporation, you could more holistically look at how injury location alters different regeneration outcomes. Yeah. Any other questions?
Oops, sorry, I was muted.
Okay.
Um,
I have a quick question
yeah,
if I
yeah, go
can.
ahead.
So first of all, congratulations for the model. I think it's very interesting, and thank you for sharing in this presentation. I was curious about when you create the PIF files from the histology images, you consider any any kind of or in this process you associate a cell ID to the different pixels, or you just consider the each pixel in the image or in each voxel in the PIF simulation as a individual entity?
Yeah, that's a good question. And that's also kind of like when we were first creating the model, it's like an interesting consideration of like how to define things because, when we read into MATLAB, it's taking like pixel by pixel, like, okay, here is like this pixel is ECM and this is fiber. So within CompuCell, you can actually see in this like older image, the like red green one. Each of those like individual like borders is showing the fiber cells in CompuCell. So biologically in CompuCell are kind of different, right? So biologically, like that whole like circle is a fiber cell, but within CompuCell, the fiber cell is actually made up of a bunch of little fibers, which like biologically would be more like myofibrils, but that's not the point. Too in the weeds. But yeah, so like the fiber cell is actually like a compartment cell within CompuCell. And so that compartment can like, and the reason for that is when there's like damage, I didn't necessarily want it to be like a whole cell had to be damaged. So by doing like a compartment where like each of the little like voxels is representing like an individual fiber cell to make up a whole fiber, it allowed me to do like smaller clumps of necrosis and to have a little bit more control over like the percentage of damage while still representing it as like a fiber cell. Does that make sense?
Yep, thank you.
Yeah, of course.
Got another question in the chat
Yeah,
here.
cool.
What are you preserving during the damage? Yeah, sorry, I looked you.
Yeah, so what are you preserving during the damage? Because I see it completely recovers in your simulation. Is it because of the ECM preservation? That's a great question. Yeah, so this iteration of the model, it was constrained to it could only place fibers essentially where there were fibers at initialization. We have since like kind of refined that a bit further. So now when we do simulations, because our contact energies are set such that it can kind of account for changes, we actually can get hypertrophy and like structure changes across the simulation so that the initialization doesn't dictate like the cross-sectional area at the end. So because biologically you wouldn't get necessarily the exact orientation that you had at the beginning. So this iteration of the model hadn't quite incorporated that yet because some of those contact parameters were added a lot more unknown parameters that had to be tweaked. But yeah, so that is something that has been updated since this iteration though. And that's a good catch that, yeah, the damage would not necessarily um, create the same architecture at the end.
I have perhaps maybe a naive question.
Yeah.
My background is not in bio at all. On slide 9, sorry to make
Okay.
you back all the way up.
You're good. I will probably do just get back this way. Okay. So
Yeah, so you're so maybe I missed something here. Is this where you were deciding where to damage the fibers? I guess so my base question is how do you decide where to initiate damage?
yeah, so it was actually it's a randomized thing. So like you would put in whatever percentage damage you want, and then it just stochastically decides where the
Okay,
damage would occur.
gotcha.
And then so like the other thing about it, um, this iteration we kind of went through a couple of different ways to induce necrosis. So the first time that we did it, it just like sporadically placed necrosis wherever. Um, Not necessarily how it occurs. So like this version is kind of showing you would get more of like a radius of necrosis. So like experimentally we would do like an injection to induce necrosis. So that's why like we would see like a radius of damage like that. So that's how this is done. But then with other injuries, you could change how like it's an input condition to the model. So like if you wanted to induce a more extensive damage, it would like create a larger section as well as like remove all of these like resident baseline cells because more extensive damage would just kill any cells within the microenvironment. So it's also something that we've kind of been iterating on, like how to represent these different types of injuries because there are several different like factors that go into it.
Mhm. Yeah, cool. That answers my
Yeah,
question. Because yeah, I was curious about like if putting the damage in different places would represent different types. So
yeah, yeah.
gotcha.
Um, so I see another question in the chat. Could you elaborate on how you chose what to include in the model? Because usually my problem is in the literature review stage and knowing the important things I'm missing. Yeah, that's a super great question and something that I think is really hard during the development process because I was always inclined to add everything. So like when I came to the hackathon, I was like, there are 27 cytokines that I need in my model to like accurately represent all of the dynamics. But like feasibly, like you're limited in terms of like the simulation in this state takes about 4.5 hours to run. So like if I were to continue adding like diffusive fields, it would just be impossible to like calibrate and like really utilize. So for me, I like went back to what is like the question that I'm asking and like what do I need for that question to be answered? Like what dynamics are occurring? So like I would always kind of revisit that question because there will always be more literature that you can include. And so really like at least for the framework, just like getting a base that's like functional with like minimal rules is like a great starting point. And then once you have that function, you just like slowly add in things that you need to answer more questions. So like never like trying to add in too much, but also keeping the framework general enough that more can be added. And thinking of ways that like will help you in the future in terms of like how you set things up to make it easy to edit for both yourself and for future people or students that may want to use it after you. Um, I know that's like kind of a hand wavy way to answer it, but that's kind of what I would say. Just like start with the basics and like outline the steppables that like you know you have to have and then like the framework that needs to be within each of those steppables. So like when I did my first version of the model, like I didn't even have all of the cell types. I don't think I included fibroblasts to start with. And I didn't have all of the different phenotypes of like the satellite stem cells and macrophages. Like I just had it very basic until like I kind of had a better grasp of how to do things in CompuCell and how like the outputs would look and then what I would need after that. Yeah.
Yeah, and Ellie, you've hit on a good point there. This is probably what we would consider like the core skill of computational modeling is being able to ask meaningful questions that can be answered with models and then determining, like you're saying, what needs to go in order to answer those questions. Learning how to use individual modeling platforms is more of a technical skill, really, in comparison to these sort of larger questions of what to include in your model. And like Megan said, often we have to give these kind of hand wavy answers because it that skill comes with sort of some practice of implementing a number of different models over time. But hopefully by the end of the next couple of days, you'll have a good start on that. So
Yeah. And I would also say like sometimes it gets a little tricky when like I feel like there's always contradicting literature, and then you're like, well, what do I include when these things don't actually agree? Or when like I feel like when I started, I was very particular, and I still kind of am. Like when building the model, I only wanted like, mouse studies with a very particular type of injury and only like immunohistochemistry, like cell counts, and no like others. So it gets like you really can like narrow yourself into a hole. So sometimes you need to be able to at least like understand the experimental data enough to see like, okay, is this representing the same thing that I'm trying to show, or is like, can I use RNA like mRNA data, or is that showing something different? Like just kind of having enough of an understanding of the literature to know like what is feasible to use to pull rules from and what maybe it's not.
Mhm. Yeah, and this point of like the model prototyping approach is important too. Like you want to start simple because the more complex your model is at the beginning, first of all, the harder it is to debug when you run into issues, but it also affects your ability to check validity if you've got a lot of interacting pieces out of the gate.
Yeah.
So, that kind of starting simple and building up process helps with that process of determining what you need from literature.
Yeah, setting up like a nice priority list of like the must-haves and the like would be nice. That
Mhm.
helped me out.
Any other questions for Megan about the model itself? We do have, while people are maybe thinking about other questions, we do have a little bit of extra time. I was wondering, because you mentioned, Megan, that your steppables are a little bit complicated.
Yep.
I was wondering if you might be able just to show that to the group for a minute and just kind of just talk through what the
Yeah.
steppables do. It doesn't have to be in huge detail, but
Yeah, I would love to. Okay, let's see. Let me just change my share. Okay. Can you see the steppable code?
yes.
Okay. So I don't use Tweedit. I moved away from that, but I remember the Tweedit days. Um so this is actually the full steppable file. So for me I have like all of my core variables set up here. There are quite a few. So within that, I then also have all of my different like conditions that I can essentially these are like on-off switches for different conditions. So like all of those perturbations were set up such that it would be like an easy on-off of like, okay, I'm gonna do this neutrophil depletion. It edits this variable or that variable or like different cytokine perturbations. I'm gonna deliver this much or that much. So then it's not something that I have to like go down and like find in my code to edit every time. And then there are certain functions that I use a lot, so defining those up at the top. Um, and so this is kind of like my importer. It doesn't get used often. It's just if I have like a new initialization file that I want to use, because you can like it's good to test different like histology images to make sure the model works kind of with different initialization conditions. So that's kind of all there for whenever it's needed. Let's see. And so more initialization in terms of like different dictionary properties that get used and how those get set up. So like I said, like for the ECM, we have a collagen that gets initialized and then is edited throughout, setting up a bunch of different like target volumes for all of these cells at initialization. And then I have my necrosis steppable, so kind of like we talked about before. So it picks a random spot for the injury to occur, um, and then it creates this like radius of necrosis. Um, and then so it's essentially taking the fiber elements and turning them to a necrotic cell type. Um, and then the neutrophil steppable. So this is, where we recruit the neutrophils. depending on the amount of necrosis remaining, and then finding the different capillary locations to decide where that neutrophil is going to come in, because it's going to come in at a location of a capillary. It also accounts for the different cytokine levels to dictate the order of capillary that it's going to come through. Set up all of the different secretions, as well as like tracking how much it phagocytoses. Then we have our satellite stem cell steppable. So all of the different dictionary properties that are helping dictate the different phenotypes and like if it's dividing, how long it's been dividing and things like that. As well as some of the aspects of how it fuses, setting up all of the different fields for that, getting it recruited. And then once it's recruiting, set up all of the proper dictionary properties. And then we have all of our different actions that we kind of talked through in the presentation. So dividing, and then when it divides, it's removing these different factors that were used. So like TNF, VEGF are all required, so then it will remove that. Kind of simulating like ligand receptor binding, essentially. Same thing with differentiation. And then this is where we can have them fuse to a fiber. So it's checking the ECM. It can also fuse to another myocyte. So you can see that the code is dense. This didn't happen overnight. It was a lot of iterating for years, apoptosis. And then the satellite stem cell division, we also have in a separate steppable. So if that signal is there, it'll then enact that steppable. Same thing with macrophages. So a lot of it also gets kind of repetitive. So like once you've done it like a couple of times, you can then leverage the code that you've written to just put it into other steppables and make changes as needed. And then it makes it go by a lot faster because then like, like, okay, this agent does this and I need this agent to do a similar behavior, but maybe tweak it this way or that way. And so that's kind of things start to like speed up once you have like a handle for the different aspects of the code. And then I also, of course, have a plot steppable. So if I am running it locally, kind of like this, I could, also put up plots. Typically, like if I'm running it on a cluster, there's no reason to have plot steppables up. So that's the other aspect of CompuCell. So when I was doing calibration, everything was run on a computing cluster. So I had a file steppable, so then I could, when it's running on the cluster, it's then going to output all of these different metrics that I need to read into CaliPro. So everything gets kind of saved into files every so many MCS, and then I can use that for calibration. And then the other aspect of that as well is under, I have like a past value steppable. So this is where I was doing a lot of my calibration. So I would essentially take the different unknown parameters and then I would pass in whatever parameter set from Latin hypercube sampling. So then that would get passed in within the cluster to update all of those different parameters. And so this is another thing that in terms of like steppable order, this matters a lot. So if I'm like calibrating and passing in parameters, that has to come early so that it has the correct parameters. So that's kind of a little bit of into the weeds of the calibration aspect on that. And then some of the microvessels, so how it undergoes angiogenesis, is all occurring within this steppable. And then lastly, fibroblasts, and I think a mitosis steppable after that. Yeah, so lots of lines. GitHub was helpful for this. I would suggest that if you're going to get code that's going to be really beefy. Because there were many times that it was super broken and it had to be backed up. But yeah, I know that's kind of a lot, but hopefully that gives like some hope for the hackathon that like this was built during, not all of it, but like, the framework was built and then it's come up to this like monster of a code now.
I was going to ask you, did you write all this yourself?
No, I mean, so Lexi and Tian, who I met at the hackathon, we all
Okay.
kind of wrote yeah, it was joint effort.
Yeah,
Um,
Galaxy is the first time I've seen a 3.9K line code. It's especially
yeah,
uncommon for CompuCell. It's not often that we see huge programs like this, but it's impressive.
yeah, I would say it doesn't like to run locally a lot, but it does great on the cluster. So that might just speak to like the bandwidth of my computer.
Hmm.
But yeah, I mean, it's doing great.
Yeah. Oh, I'm sure this would bring my computer to its knees. So I have one more question, then I'll let other people ask some.
Yeah.
You were mentioning that you do have ECM in this simulation. That's something that we often get questions about different ways of representing the ECM in CompuCell because there's not a native like ECM plugin that I know of, at least in CompuCell. So I was wondering if you could talk a little bit more about how you built the ECM into your simulation.
Yeah, it was kind of like a weird thing conceptually because I feel like when I came in, I was like, oh, agents are cells. So like I wouldn't represent the ECM as a cell because it's not a cell, it's like a structure thing. But within CompuCell, essentially everything is just a cell. So like within yeah, so this kind of like shifts a little bit. So like I have like essentially a fiber layer, and so that's where I have like my ECM cells, and like my fiber cells. Well, yeah, fiber cells, and then like the capillary cells and all of that. So it is represented as a cell, but for when initially started, it was a frozen cell. So like it couldn't move or do any migration. It's not, I don't think it's frozen anymore, but like the contact energies are set such that it's essentially not going to move.
Hmm.
Um, and so that's kind of like I don't have medium within the simulation, really. Oh, I guess maybe kind of in the cell layer, but like for the like microstructure layer, it's like all ECM. And that's kind of like my medium of sorts. And so that's what like the cells are like migrating through. So the other thing that like in efforts to keep all of the like cells within the ECM, I have another like diffusive field that's just like repel. So like the fibers will just secrete repel to keep things like within the ECM so that like they're migrating, not on top of other cells. Yeah, so that's how I chose to represent it. I feel like there are other ways that you could do it, but like for this scheme, it was kind of the thing that worked best. Um, yeah.
Hmm. Yeah, and when you say that you have them on different layers, you're talking about like Z slices in the
Yeah, so
simulation.
originally, yeah, so this one, yeah, is on layer 0 and this one is on layer 2.
Yeah. Okay.
Um, and that's just because like CompuCell wouldn't let me have like cell on cell. So like satellites, yeah, it just wasn't gonna like work another way.
Yeah, that's using Z slices for different layers in a 2D simulation is something you can do. We haven't really talked about it much this week. It may come up, I think, in um, I think one of Julio's sessions later this week. But yeah, that's super interesting. The just modeling the ECM underneath the cells, basically.
Yeah. And it works decently well. The only like other consideration is when something gets secreted, I keep it all on layer 0, just to have like, because there gets to be like a mismatch if like the cells are secreting on layer 2 and then the fibers are secreting something on layer 0, like all of a sudden they're not talking to each other super well. So like everything that the cell secretes, like I have a user-defined function that like makes it like on layer 0, just to like
Mhm.
consolidate some of that. Um, yeah.
Yeah, cool.
Yeah,
Yeah,
and
so I think
then oh, thank you, Yasmina, for putting in the publication. I'm also happy to share the slides if anyone wants those too. But yeah,
yeah,
everything
we'll
I think I presented on is in the publication too.
yeah, I can you can send me the slides if you already haven't, Megan, and I can get them uploaded to the drive for people to download, and we can also add a link into the GitHub code as well if anybody wants to attempt to download and get set up to run it if they would like to try to run it on
Yeah,
their
yeah, I
own
can do that.
systems.
Cool.
Yeah, but like Megan was saying, this one apparently takes a while to run, so it may be something that you don't see a whole lot of progress on at first. If you do try to run it locally, you might have to let it sit for a while.
Yeah, it's one that like I don't know, like save the screenshots and then let it run overnight and then like look through it.
Yeah. So that also gets back to Ali's question of there's also this, in addition to deciding what is realistic, like what's valid to include in the simulation, there's also always this question of what's going to make my simulation computationally infeasible. When I was doing my
So,
master's, I was doing protein folding simulation, and that's the kind of thing that you have to set up and then send to the cluster for a month and hope that hope that when you get it back, it's useful. Um, so you do have to keep that kind of stuff in mind too, because you can make it as fine grained as you want, but the finer grain it is, the more typically the more computationally expensive it'll be.
yeah,
Um,
I know for us, there was also talk of like, I'm doing like a very small subsection of a muscle, right? So like at one point we were like, what if we do the whole cross section? And just to initialize the model, like it was not feasible to run something like that because the number of cells too, like CompuCell just couldn't handle that many cells. Yeah. So
yeah,
I mean, we still are able to answer our questions with our little section, but it is something to consider.
right. Yeah, we had some super high resolution protein models that was like, so how long will it take just to relax the system before we can start? And it was like, oh, okay, just the relaxation takes a month. So it's like, can't do that. We need to we need to simplify this. So
Yeah. Or the other thing that we discussed was like, should we have like a steady state for some of our cytokines? And like, how long do we run the simulation before injury to reach a steady state? We ultimately just decided to run the steady state as an initialization and then load that in. But all of these things are just like considerations for like, how long do you want your simulation to take? Because especially when you're running like a ton of like perturbations to test different things, and then considering the stochasticity if your model needs that for like biological relevance, then all of a sudden you're talking about like thousands of simulations. And then if you can't, like you would have to just be able to run that on a cluster and have it run in parallel. Otherwise, I would have been in my PhD for like 20 years. Um,
right. Well, cool. So any last questions for Megan before we break here? All right. I think you can probably call it quits. We'll get an extra 15 minutes of break here for people that are coming back to the workshop. So I do want to remind people that we will reconvene in the main workshop meeting room at 4:30. I'll be sending an email out in 15 minutes with that reminder in there. So we will go ahead and say thank you to Megan again for a great presentation. And we can go ahead and break. So again, those people who are coming back, we will reconvene and start module 4.4 on initial cell layouts. So Megan did talk about using a PIF file for the original setup. We actually have a module at 4:30 on how to do those and how to make those files. So it'll be a good follow-up for what Megan talked about here. So we will
cool.
see everybody back in the main session at 4:30.
Thanks. Have a good one.
Bye. Thank you, Megan. And I'll be in touch about getting the slides.
Sounds good. Bye.